Bismillah. Assalamualaikum warahmatullah. You're listening to microbiology lesson on microbial growth and control. In this part 1, we will focus on two learning outcomes. First is for you to be familiar with some basic definitions and concepts related to the microbial growth and control. And that also means being able to discuss concepts related to cultivation and measurements of microbial growth. Now, the best place to start is the definition of the word growth itself. If you notice, we use the word growth quite differently in microbiology compared to when we talk about macroorganisms or larger organisms. So in microbiology, when we say growth, we often don't mean an increase in size. Rather, growth means increase in numbers. So that doesn't mean microbial cells do not increase in size. They do. But generally speaking, when we conceptualize growth for microorganisms, we think about how many they are instead of how big they are. Another important terms you'll come across is the ending "phile" and "tolerant." When technical terms end with "phile," it means the microorganisms like it or need it. So, for example, aerophile means the organism likes oxygen or needs oxygen. On the other hand, aerotolerant means the organism doesn't like oxygen, but they can survive it, they can tolerate it, and they can grow within that environment. Now, since growth is defined as an increase in cell numbers, cell division becomes crucial in microbiology. It might not feel that way now because you have learned that cells divide even from when you were in school, and by now you might have taken it for granted. But cell division is an event that is so fundamental in life, at least the biological aspects of life. So people do PhDs on various aspects of cellular division. So two general ways in which microbial cells divide themselves is binary fission and budding. Binary fission is when a cell makes a second copy of its components and then split into two cells. So many of your common bacteria do that, E. coli, Staph aureus. Budding, on the other hand, is when you have a new cell develops on top of the old cell. Budding is a more of a fungal thing. So like yeast use budding to divide. But some bacteria do budding as well, some cyanobacterial species. When a cell is dividing, we call it a parent cell or a mother cell. One of the first things that happens to a mother cell is the elongation of nucleoid. Her nucleoid becomes longer. A lot goes on in there, replication of genetic materials primarily. At some point, the nucleoid splits into two. You have two nucleoids that are genetically identical, at least theoretically, because sometimes mutation occurs. Anyway, after that, what you'll see is the formation of septum or partition. So this septum will occur along the transversal plane, meaning the line of partition is always in between two nucleoids. And once the septum is complete, you'll have two daughter cells. So notice that what we call the daughter cells are the original mother cell which split in two. So you won't have one mother cell and two daughters exist at the same time. So one cell becomes two, two cells become four, and on and on. And we can plot the numerical changes of microbial cells mathematically on a graph. So this graph is called the growth phase graph. On the y-axis, you've got your number of cells. We express it in logarithmic value instead of linear value because of the doubling of numbers, meaning because the cells don't grow from 1 to 2 to 3 to 4. It grows from 1 to 2 to 4 to 8 to 16. So that's the pattern. Now on the x-axis, you have time. Any units of time will work here depending on the rate of growth of your microorganisms and experimental conditions. Sometimes it takes hours, sometimes it takes days. Now, this graph is special. You can literally draw this graph and show it to any microbiology student in England or China or Nigeria. They all know what it means because this graph is a pictorial summary of what happens when you grow bacteria. When you grow bacteria in a fresh medium, in the beginning, you have lag phase. In lag phase, it seems not much is happening in terms of cell numbers, but intracellularly, a lot of things are going on. The cells absorb nutrients from the medium. They synthesize enzymes. They produce ATP molecules. Once they are ready, the cells switch into the log phase. And in log phase, you'll see an exponential growth. So for example, 1 million cells can easily become 1 billion cells within several hours. However, they can't grow in the log phase forever. At some point, the growth will switch into stationary phase. In stationary phase, the number of live cells becomes constant, so the graph flattens. Now, mind you, it doesn't mean the cells stop dividing. So it's just the rate of new cells being born is the same rate as older cells dying. They die because of two things. One is because the medial nutrient is depleting, meaning the food is running out. And two is because the waste products are accumulating. So the environment becomes toxic. At some point, the situation becomes so untenable that the microbial growth switches into decline phase. In the decline phase, the number of live cells fall. So this is the apocalypse. So you're going to see lots of malformed cells. Their shape gets so messed up that you can't identify them anymore, and everyone is dying. Well, not everyone. In species that can form spores, you're going to witness hope here. You'll see the development of endospores and free spores. And the spores may survive even for years, waiting to be revived in the future. Other concepts that you want to keep in mind when you research microbial growth are generation time and the difference between synchronous and non-synchronous growth. Generation time is the time for a microbial population to double in numbers. So if your bacteria double from 1,000 cells to 2,000 in 1 hour, that means its generation time is 1 hour. Usually they are under an hour, but there are microorganisms that takes almost a day to double their numbers. So sometimes at uni, you come across postgrad students who sleep in the lab. You saw them, this morning wearing the same clothes they wore yesterday evening. They do that not necessarily because they love science so much that they don't want to go home, but they might have to do it because their microbial sample has a short generation time. So for example, they need to profile the microbial growth. It means they need to take spectral readings, for example, every hour for 24-hour period. So they can't stop. So they have to sleep in the lab. Now, this one is interesting. Before you study microbiology, you might think science gives you the direct, objective truth about reality. Now, as you study deeper into science, you will realize that's not true. For example, I taught you that if you have 1,000 cells per mil in the first generation, you'll get 2,000 cells in the second generation, then 2,000 becomes 4,000, 4,000 become 8,000. If you visualize it on a graph, the line looks like a staircase. So it assumes that cells synchronously divide. So this theory is called synchronous growth. Synchronous means together at the same time. So the theory implies that all 1,000 cells divide together at the same time. But that is not objectively true. We teach you that theory to reduce objective reality, to simplify it for you. In reality, cells don't divide at the same time together. Each cell divides at different time duration. So it is called non-synchronous growth. And if you plot that assumption, you have a more smooth slope upward, not a staircase. Now, when you hear technology, don't only think of machines. So technology is the application of scientific theories. So that includes techniques as well. For example, aseptic principle is a set of techniques to prevent contamination. So that is technology. What techniques you use depend on the context of your procedures. For example, for a simple culture transfer in your lab class, you just need to establish a sterile zone close to a Bunsen burner. But another example is for a surgery, like the ones you've seen on Grey's Anatomy or other medical TV shows, you need to scrub your arms. You need to wear a full surgical gowns and scrub caps. So it's more elaborate. In your context, the reason you need aseptic techniques, among other things, is to get what's referred to as a pure culture. A pure culture is when you have only one species in your culture. One way of knowing this is the colony on your plate shouldn't have any variance in their morphology. They must all look the same. On solid media, you get a pure culture by strict plate method or pour plate method. We'll talk more about it in a minute. But the purpose of either method is the same. You want to isolate individual colonies from a mixed culture. Then, once you have isolated pure cultures from it, you can run further diagnostic steps to do biochemical or genetic tests to find out their identity. The media we use to cultivate those microorganisms can be categorized in a couple of ways. One is a natural versus synthetic media. Natural media are the ones you have taken from nature as is. For example, your natural media are the soil or the lake water where you took your microbial samples from. Most of the times, though, in the lab, we don't do that. We use synthetic media. These are ones made in the lab with components that mimic the nutritional requirements of our microorganisms. The synthetic media has an advantage over natural media in terms of replicability. So it allows other scientists to replicate the conditions in which we grow our microorganisms. And that is especially true when we use chemically defined media. So these are the ones where you know precisely what's in them, how much sodium, how much calcium, how much ammonium chloride. Now, at times, chemically defined media don't work for certain microorganisms. So in those cases, we can use complex media. Complex media doesn't mean we don't know what's in them. We do, but it is less defined. We don't know exactly how much chemical components in each batch. We only know that the media have, for instance, peptones or blood from animals, not the precise chemical compositions. But for day-to-day lab work, a more useful categorization of media is the one based on their purpose, based on what we want the media to do. For instance, we use selective media to select certain types of bacteria to grow, while inhibit other types of bacteria that we don't want. One medium that do that is called Levine EMB agar. That agar selects for gram-negative bacteria, so you're less likely to have gram-positive bacteria growing there. What's cool about EMB is if you have E. coli on it, the colony will have this green metallic color. So you can differentiate bacteria on EMB as well as use it as a selective media. So those media that can help us differentiate bacteria is called differential media. So a medium can belong to two categories at the same time. So beside EMB, you also have, for example, McConkey agar. So this agar has a pH indicator as part of it. The media becomes red when it's acidic. And if you have lactose fermenting bacteria, they will also show up as red colonies. That helps you differentiate lactose fermenting bacteria from non-lactose fermenters. Sometimes you're not looking to differentiate bacteria, you simply want them to grow, and they can become fastidious, which means hard to grow, very choosy with their food. So for those microorganisms, you can use enriched media like chocolate agar. It's not actually chocolate, by the way, it's animal blood. So don't think about licking it or something. So ask your lecturer to buy a proper chocolate later. For some microorganisms, the issue about growth is not just about what they eat. Sometimes it's about what they breathe. So for some of them, oxygen is lethal. So on top of choosing the right media, you have to grow them in an anaerobic chamber where you deprive all oxygen in their environment. Once you've got your microorganisms cultivated, you may need to measure them. For measuring microbial growth, one of the techniques you'll commonly use is serial dilution. The reason you want to serially dilute your samples is because sometimes when you grow them on a plate, you have too many colonies to count. So what you do is you take 1 ml of your sample in the original test tube and you add that to 9 ml of your diluent, your broth media. So what you have then is 1 in 10 dilution. And when you do this, theoretically, the number of microbial cells is diluted as well. If you have 330,000 cells, if you dilute it to 1 in 10, you will have 333,000 divided by 10, and you'll have 33,000 cells per mil. What you can do then is repeat that step and make it 1 in 100, 1 in 1,000, or to whatever dilution that you need. And of course, correspondingly, your cells will be diluted at the same dilution level. The higher the dilution, the lower the number of cells per mil you will get. And for any of the diluted samples you've got, you can start measuring how many bacteria you have. The first way of doing this is the spread plate method. You take 0.1 ml of your broth. You take it from one of the dilution tubes. Then you pour it on a solid agar and you spread it all over the surface. The second way is pour plate method. So you'll take 1 ml of the sample and add it with 9 ml of melted agar and then pour it into an empty plate. Of course, you don't do it just on a single plate. You need replicates. So you repeat it for, say, 3 plates, then you start counting your bacteria. Now, this is important. Notice that you don't directly count bacterial cells with this method. You count bacterial colonies, or more technically, you count observable units that the colonies have formed. So that's point number 1. Point number 2, scholars of this discipline have like an ijma or consensus regarding the number of colonies you should count. So they say you only include your result if it is between 30 and 300 colonies. Anything fewer or more than that range, we don't use it. And then whatever X number you get, you average it based on the number of your replicates. So if you use 1 in 10,000 dilution sample, and then you get 34 CFU for plate 1, 33 for plate 2, and 32 for plate 3, you'll get an average of 33 CFU for your 1 in 10,000 dilution tube. And now that you know you have 33 CFU per mil in that tube, remember, this is colony forming units. It doesn't mean you have 33 cells in there. Yeah. So now you have 33 CFU in your 1 in 10,000 dilution tube. Knowing that you can just use simple math, you can calculate yourself back to the concentration of your original sample. So 33 times 10,000 equals to 330,000 CFU per mil. So it's that easy. Now, for sure, counting colonies is not the only measuring methods. As you take more advanced courses later, you learn, for example, hemocytometer. So it's a slide that has etched grid on it. So we use it, for example, when I did my postgrad in immunology, I use it to count leukocytes from lungs. So if you've got asthma or flu infection, you're going to see increased number of leukocytes in the lungs. So that's one example when you can use hemocytometer. There's another method. I've never used this one. It's called most probable number, or MPN. You make a serial dilution, then instead of plating it, you cultivate your sample in tubes with durum in it. Durum is a tiny tube that you can put inside a test tube. So you put durum upside down to trap CO2 produced by the cells. What you do is you put 5 tubes containing 10 ml of that sample. Then in the second group, you put 5 tubes with 1 ml of that sample, plus 9 ml of media, of course. And then in the third group, you put 5 tubes containing 0.1 ml of that sample, plus 9.9 ml of media. Then you grow them overnight. And then you count the number of tubes that has gas trapped in the durum tubes. For instance, for the 10 ml group, you get 5 positive tubes, meaning 5 tubes with gas trapped in each of them. For the 1 ml group, you get 2 positive tubes. And for 0.1 ml group, you get all negatives because it's too diluted. Then you plug that number into the MPN index that you can find online. The MPN index is a long table that says if you get 0 positives in all 3 groups, your MPN value is less than 2. At the other end, if you have 5 positive tubes,
For all three groups, it means you have over 1,600 MPN. So you go through that index and you find the one you've got. In this case, you find 5 positives for 10 mil, 2 for 1 mil, and 0 for 0.1 mil. So in your case, you report your MPN value as 50. Another measuring method is, instead of counting cells, we quantify the turbidity of our liquid samples. We quantify how cloudy it is. The assumption here is the more cloudy your sample is, the more microbial growth you've got in there. You quantify the turbidity using machines such as spectrophotometer. What a spectro does is it shoots monochromatic beam across space, and the sensor on the other side will record the amount of light that reaches it. So if you put a very cloudy sample in the middle, much of the light wave will be absorbed by the sample instead of the sensor. And then the machine will compute it and give you a value called absorbance. So that value represents how much bacteria you have in your sample. All right, this is the last point. Sometimes your bacteria just stop growing, even if you provide them with a culture condition in which they grew before. In the past, microbiologists just assume your bacteria is just dead. But today, we know it's not necessarily the case. Bacteria can get into what's called a VBNC state, which means viable but non-culturable states. This become an issue, especially in food microbiology, because food processing can trigger this phenomenon. For example, there's a paper that shows if you filter an orange juice in a certain way, that process injures certain bacterial cells in that juice. And that injury can trigger VBNC. So if you try to culture the bacteria from that juice, the bacteria won't grow. So you thought, cool, the bacteria are dead then. But when you use another machine, like flow cytometry, you'll find that the bacteria are not dead. And they are still viable, even though they are not culturable. So that's an example of VBNC. I'll leave you that paper and some links below if you'd like to explore more about it. Okay, that's it. I pray that you've learned something useful. Talk to you in the next one. Barakallahu li wa lakum. Assalamu alaikum wa rahmatullah.